Alrighty, hi guys. Um, this is the third installment of the um, Solving Problems in Mendelian Inheritance series. This one's for week 7, where we cover all the exceptions. So last week we taught you all the rules for how Mendelian inheritance works in general, but there are a lot of things that aren't explained by Mendelian inheritance principles we taught you last week. For example, according to Mendel, we should only have 2 skin colors. We should have extremely black people and extremely white people and nothing in between. But we don't just have 2 skin colors. We have an entire gradient. According to Mendel, we should only have 2 heights. According to Mendel, there should only be 2 flower colors. You should never get spotted flowers or pink flowers or anything like that. So last week we taught you the rules. This week we're going to teach you the exceptions to the rules. But to be honest, this week's not nearly as bad as last week because once you know the rules, these are just slight variations on it. They're just little twists to the story, but the Punnett Squares work exactly the same. It's just what the Punnett Squares means slightly changes somewhat. But if you understand the idea of Punnett Squares from the first 2 videos, you're home free on this week. It's not going to be as bad at all. There's just a couple little tricks that we have up our sleeves that make these ones a little tricky if you don't know what you're doing. So the idea of this video is to go through each of the different kinds of inheritance and tackle a problem on most of them at least, and explain the principles behind it so that you get a greater understanding, maybe to clear things up on how to solve these kinds of problems that you'll run into. This is that homework from this week. It's 5 problems long, and there's a couple of different principles here. I'll go through 7 different kinds of problems. So I'll cover these 5 first, these type of problems first, and then I'll knock on the last 2 principles at the end. So we've got, here's the 7 different things to go over for this week. So we'll go over a lethal allele problem, and then we'll hit some an incomplete dominance. We'll go to codominance. Then I'll cover how to tackle blood type and multiple allele problems and sex limited, and then we'll go to the idea of polygenetic inheritance. Sounds good? Okay. So lethal alleles, this is exactly like the Punnett Squares you're running here are exactly like the Punnett Squares we did last time. So here's a lethal allele problem. Achondroplasia is an autosomal dominant dwarfism disorder in humans caused by a mutation in the FGFR3 gene. A heterozygous individual is a dwarf. Individuals who are homozygous for the dwarf allele miscarry before birth and are never found in the population. What are the odds that a child born to 2 dwarfs will also be a dwarf? Okay, so now here's the interesting thing about this. So dwarf, we told you that the dwarf is heterozygous. So just as a remembering from last week, heterozygous means they've got one of the big allele and one of the little allele, or one of the dominant, one of the recessive. In this case, I didn't tell you which one, but it also doesn't matter because dominance and recessiveness only comes into play if you're in the heterozygous condition. In this case, I told you what heterozygous was. I told you it's yellow. So I guess in this case, if we had to nail a dominant one, or sorry, not yellow, it's dwarfism. So in this case, it's autosomal dominant, which means this is it's on an autosome. It's on the chromosome 1 through 22, I don't care which. And it's dominant to the normal condition. But if you've got 2 of the dwarfism alleles, the baby never gets born. It's impossible to survive. There are lots of reasons behind these. The reasoning behind lethal alleles is on a couple of these more key proteins. If you've got one copy that works, then that one copy that works can still keep things running fine and dandy. It's when both of them fail that it's a problem. For example, like if you have a fish tank and there's 2 filters on the fish tank, then if one of them gives out, you've still got another filter on the fish tank, and so the fish tank's not going to get dirty. But if both filters give out, then the fish tank doesn't have a chance. So the idea here is when it's in the heterozygous condition, sometimes you won't notice it. Sometimes, in this case, it does have an effect, but they're still able to survive. But if both of the genes are broken, the FGFR gene makes the FGFR3 protein, which is very essential. We'll learn about it in week 9. It's very essential for making the body function right. So if you have 2 broken copies of it, you can't survive. That's the theory behind it. So now with a Punnett square, so we have 2 dwarfs. So we told you up here that the dwarf means they're heterozygous. So I'll use R because that's one of my favorite letters here. We got a big R, a little r, carries a big R and a little r, because I told you both parents are big R, little r. Okay, so if you run your Punnett square, this is the standard 25, 50, 25 gig. We got, okay, here we go. So, I mean, this is exactly like every Punnett square you've ever seen before. Now, if we ask you the phenotypic ratio here, it says, what are the odds that a child born to 2 dwarfs will also be a dwarf? Now, logic says this, we told you heterozygous means dwarf. So this looks like 50% because this quarter is dead, which isn't a dwarf, and this quarter is normal. So you can actually have a normal kid from 2 dwarfs. So what we taught you last week says 50%. Here's the catch. The catch here is being dead isn't a phenotype, it's a state of being. So the way I like to think about this one is, let's say we have, I don't know, 4 kids. Let's say there are 4 kids. Or maybe in my mind, let's go to the no, we'll go to the 4 kids. Okay, 1. So this is kid 1, kid 2, kid 3, kid 4. Okay? Or maybe let's make it 8 kids. It's an LDS family. 5, 6, oops, wrong button. 5, 6, okay. And let's pretend that this family perfectly fits the ratios of the Punnett square. Sounds good? So this wouldn't happen in real life because remember, a Punnett square says the odds for every kid. But let's just say that this family perfectly fits the Punnett square. So we would wind up with about, according to my Punnett square, 50% of them are dwarfs. So out of the 8, I would expect 4 of them to be dwarf. Say the first 4 there are dwarfs. Follow me so far? This is just, again, for theory, we're pretending that it perfectly follows the Punnett square. Okay. Then we would expect 25% of the kids to be normal. 25% of the kids would be normal, so we'd have 2 normal kids. Oops. And then, according to our Punnett square, 25% of them would be dead, because big R is dead. So these guys are dead. But can you count them if they're dead? Because they don't, because from a scientific perspective, unless we're chasing cells, you'd never know if it miscarried or not, right? So these ones, from a scientific perspective, don't exist. Does that make sense? So there were 8 pregnancies, 6 of which were born, 2 of which died, according to our Punnett square. So now, if you look at the phenotypic ratio of the children we can count, 4 out of 6 of them are dwarfs, 2 out of 6 of them are normal. What's 4 divided by 6? That's 2/3. So with these lethal allele ones, if you get 2 heterozygous individuals together, you can actually work in thirds, or 33% and 66%, as opposed to the quarters that we're normally used to, because being dead is not a phenotype, it's a state of being. So in this case, the answer would be, it was asking, what are the odds that a child born to 2 dwarfs will also be a dwarf? The answer here would be 66%. Does that make sense? Because 66.66666666, whatever is 2/3. And so the answer here is 66% because 2/3 of their living offspring, 2/3 of the children born, or for children that are born, there is a 2/3 chance that they will have achondroplasia. Because that last quarter, they're dead, they're not born, so they're disqualified. So there are only 3 boxes that matter. So you wind up working in thirds instead of quarters. So be careful on your lethal allele ones. Now it's possible on lethal allele ones to have 4 squares. For example, if this person was a little r, now all 4 squares survive. And so we're back to working in quarters again. Does that make sense? Because there's none of them got homozygous dominant there, so none of them die. So in this case here, if you had a case where none of them are 2 big R's, then yeah, you're back to working in quarters again. But in the case where it's big R, where it's 2 heterozygous individuals like this, you wind up with, there's a lethal combo right there, so you wind up working in thirds. So the answer here would be 66% achondroplasia. So keep that in mind. Don't get caught on those fractions there. So that's lethal alleles. That's the only alleles that is exactly like every other one you've worked with. Okay, here's the next one. In humans, curly hair is inherited in a pattern of incomplete dominance. Individuals homozygous for straight hair will have straight hair. Homozygous curly individuals will have curly hair, and heterozygous individuals will have wavy hair. Now, here's the idea behind this one. According to the Mendel principles we taught you last week, there should be a dominant allele and a recessive allele, and the dominant one should overpower the recessive in the heterozygous condition, right? So in this case, if having curly hair is dominant to having straight hair, then a heterozygous person should have curly hair, not wavy hair. But in this case, neither allele is strong enough to overcome the other one. Neither one of them is completely dominant. And so they actually blend together, and you get kind of a compromise between the 2 alleles, which is wavy hair. Straight the blue, you get wavy. Other examples that they love to use are flowers. They say red is dominant to white, and heterozygotes are pink. Pink, that wasn't an option. We have a red allele and a white allele, but when you put them together, you made a new phenotype because we blended it like paint buckets. So the way that I remember incomplete is paint buckets. It's a blend. We create a new phenotype that didn't exist before. There's a new color, or a new, in this case, a new physical characteristic that didn't exist before. Wavy hair wasn't an allele. There wasn't an allele for wavy hair, but because the 2 alleles blended together, because neither one was strong enough to beat the other out, you got wavy hair. Does that make sense? So down here, if a straight-haired man, so if he's straight-haired, and let's just say, I didn't tell you which one's dominant, which one's recessive, because neither one's really dominant. So I'll just use little r for straight. So over here, I've got little r equal to straight, and big R is equal to curly. Okay. We've got, so, and then big R, little r, of course, is wavy. So we have a straight-haired man. If he's straight-haired, that means he has to be 2 little r's for wavy or wavy. Down here, he marries a wavy-haired woman. So she's going to be big R, little r, because it's incompletely dominant. So if you get both alleles, you wind up with a wavy phenotype. We do our Punnett square here. So again, the Punnett squares function exactly the same. It's just figuring out what the Punnett squares mean that slightly changes this week. All right, so here's the Punnett square. So what are the odds their first child will have wavy hair? Phenotypic ratio here those two will have wavy hair, and those two will have straight hair, because little r means straight hair. So we would have a 50% chance of having a kid with wavy hair. Now notice this isn't curly hair. The big R doesn't dominate over the little r. The heterozygous, they blend together and make wavy hair. That's the difference here. So again, the Punnett square didn't change. It's what the Punnett square means. In this case, what the heterozygous means changes. So in this case, it would be 50% wavy, 50% straight. Now notice you can have these kinds of story with, you can have questions like this with X-linked alleles as well. In fact, your Calico Cat problem was something very similar. It's actually going to be the next principle I talk about. That's called codominance, but it runs off the same principle. You remember last week, we had that really obnoxious Calico Cat problem. I think it was 5, where the heterozygous females were had black and yellow. Same case here, except instead of black and yellow spots, it's wavy hair. But that would mean a man, assuming this were an X-linked allele, a man could never have wavy hair because man can't be heterozygous for an X-linked trait. And we'll go over that here in a sec with codominance. But you technically did a problem from this week last week. That was problem 5. Here we go. This is a codominant one. So codominance, and I might even skip over this problem, but codominance is the exact same story here, except instead of the heterozygous blending together to create a new phenotype, the phenotype with a codominant one is neither allele strong enough to overpower the other one, so they both show up in spots. Does that make sense? This is the Calico Cat problem. You get spots. Because if you look up what a Calico Cat looks like, Calico Cat. See, Calico Cats, they have black and orange spots. So we had a black allele and we had a yellow allele, but when we blended them together, we didn't come out with puce orange, right? And we didn't come out with yellow. We came out with black and yellow spots. This is calico. Now, if you go back to that Calico Cat problem, so let's just ignore my chicken one here. If you go back to the Calico Cat problem that we did last week, you had, it was an X-linked allele, right? An X-linked allele, and it said that yellow was dominant to black, and heterozygous females are calico, right? A lot of people get confused with that one, but here's the reasoning behind it. Let's do the Calico Cat problem right here. We have, the man was yellow, so we will do X big B here, because yellow was dominant to black. So he was X big B Y, because remember, the Y chromosome is not an X chromosome. Don't get that mixed up. So the allele's not going to be on the Y chromosome. There's not a little letter on top of it. It's just the Y chromosome. So a man only has one copy of the allele. Female was calico. It's sad that I've done this so many times. I've memorized the question. Okay, here we go. Female was calico, and we told you in the question that was heterozygous because calico was in heterozygous, so we get spots. Now we do the cross. End up with XB to big B, so she was yellow. This one, B little b, so that one was calico, because remember it was heterozygous. Over here we had an X big B Y and an X little b Y. So he's yellow, he's black. Notice it's impossible for a man to be calico. It's physically impossible. Because if because he has to be heterozygous for the codominance story to work. The two alleles have to be competing in order to be codominant, or in order to show up as spots. And the man doesn't have two of them to compete. So it's impossible for a man to be heterozygous. In fact, it's impossible, or for an X-linked allele. It's impossible for a man to be homozygous for an X-linked allele as well, because the definition of homozygous is both of his alleles are the same, and he, again, doesn't have two. So we have a new vocab word called hemizygous. I'll even write it out. Hemizygous. Hemizygous means half. So this male up here is hemizygous dominant, is how we'd say that. Whereas if he was X little by, he would be hemizygous recessive. Does that make sense? So in this case, with the Calico Cat problem, that was a codominance problem, because you wound up with this one, rather than this female cat being yellow, she came out being spotted. That's all it is. Again, same Punnett square, just heterozygous means something different. So that's the difference between codominance and incomplete dominance. Incomplete dominance, they blend together to form a new color or a new phenotype, a new thing that wasn't an option before with the alleles. There wasn't a wavy-haired allele up here, or there isn't a pink flower allele in those red cross to white ones that you run into in your homework. Codominant, the difference is you get spots. There are patches of black and yellow, or like your traditional milk cow of black and white. Good so far? Okay. The other ones here on the list, let's go into blood type here, because you have to solve a blood type problem, and I want you to be able to understand how this works. I'm not going to solve the problem for you. I'm not even going to tackle the problem. I'm going to show you how typing works, that you understand it, because blood typing's fun. Actually has three principles involved. It's got codominance, it's got multiple alleles, which I'll explain in conjunction with this one, and it's got complete dominance. So when we say complete dominance, that's the pattern of inheritance style that we learned last week, where there is one allele that is dominant and one allele that is recessive, and the dominant one overpowers the recessive one. That's complete dominance. So with blood typing, it's particularly tricky because we got complete dominance, codominance, and multiple alleles to worry about. So what I did, so on my website, for those of you that use my website here, my website is sites.google.com/u/DNaTurFiles. It's right up there at the top there, if you want to go to it. On the week 7 page here, if you scroll down, there's a link here to download my blood typing simulator. This isn't required, but I find it as a helpful visual tool, and I'll use it here in my example on how this works. Okay, so it's going to pop up when you first open it. Actually, let me show you how this. If you download it here, okay, so it's going to show up, and I wrote the instructions on how to use it up here. So in case you forget, it's good. It's right up here at the top. Make sure to press the enable editing and also press enable content. Otherwise, all the fancy features won't work. Okay, so if you scroll underneath the instructions here, you'll come down to my actual simulator. For this class, we don't worry about the RH. So if you're familiar with blood typing, they always say your blood type is A positive or A negative or O positive or O negative or whatnot. The RH is the positive or negative. I built this spreadsheet for anatomy and physiology classes on campus, and they have to know about the plus and minus and how that works. For this class, we don't worry about that. So ignore the RH. You're welcome to toy around with it if you want, but you don't need to know about the Rh. That's a separate gene. So we're just worried about the ABO gene. So the ABO blood typing is basically the story here is your body, the immune system is trying to recognize good from bad. They want to know if a bad guy cell gets inside your body so that they know how to destroy it. So we've got a little friend or foe system of passports going on here where they poke a little sugar or a protein out the side of the cell that says, I'm friendly, don't kill me. And if I'm an invading cell, then when I poke out a flag that says, I'm friendly, don't kill me, the flag doesn't match the flags of the other guys in the body, the actual natives. So my flag, the immune system is like, oh, your flag doesn't match, and kills the cell. So it's a defense mechanism here. So what we're trying to do here, going over a table here. Okay, here we go. What we're trying to do here is determine what sticks, what flags we're posting on the outside of the cell. And this gene, the ABO gene, codes for proteins that make the flags that they poke on the outside. And there are two different kinds of flags you can have on the outside. You can either have the A-type flag. We call it the A antigen. So an antigen is just another name for the flag. So we've got the A antigen, the A-type flag, or we can have the B-type flag, or the B antigen. Good so far? So there are two possible flags that we can make out of this gene. Now the trick here is there's actually a third option. Now this whole time we've been talking about genes where there's two options. There's a dominant allele and a recessive allele, and that's it. You either have two of the dominant, two of the recessive, or one of each. In this case, we're saying there's more than two options. That doesn't mean, though, that there's more than two spots available to put the alleles. Let me say that again. That doesn't mean that there's more than two spots. We're still working with genotypes that have two alleles in them, but there's a wider selection of alleles to put there. Does that make sense? So we don't just have two options to put in those two boxes anymore. We have multiples. In the case of blood typing, there's three options. So there's the A allele that makes the A-type stick. There's the B allele that makes the B-type stick. And there's the O allele that doesn't make a stick. So the O allele is actually the broken version of the gene, so it just doesn't make a stick. It doesn't work anymore. So the O version plain just doesn't make any flags. It doesn't make any sticks. So the way this works is the remember how I told you there's a complete dominance problem, a codominance piece to this, and a multiple alleles portion. Well, there's the multiple alleles. The multiple alleles is there's more than two options that I can put in here. So I could have an AB or a BB or an AO or an OO. Does that make sense? So my genotype, there's now more than two options of things I can have in my genotype. The codominance one is the A allele is codominant to the B allele. So if you have an A allele and a B allele, you actually wind up being AB blood type. And I'll show you here in a sec. It'll make more sense how that works. The other the other one is the complete dominant, because the O allele is recessive to both A and B. Does that make sense? So if the O allele is paired with A, you don't wind up with an AO phenotype, because there's no such thing as AO blood type. It's A blood type, because the A overpowers the O, but the A can't overpower the B, so you wind up with AB phenotype. Now let me show you why. Because I find if you visualize it, you're able to understand the reasoning behind why this all works. And so the blood typing will go much more smoothly for you. Because it's actually not that hard, and in reality, it's quite fun. Okay, let me zoom back in here. The other thing for my simulator, you don't have to worry about the antibodies over here on the right. It's fascinating. You don't need to know about them. that's again for the anat and phys classes. Okay, here we go. So if you have an A allele and an A allele, you're going to make a red blood, you're going to make blood cells with A-type sticks poking out the outside. See that? So that's what the blue sticks mean. So this is called A-type blood. Again, ignore the negative and positive. With the RH, you don't need to know that. This is A-type blood. Okay. Well, if we've got, if we have, let's see, clear genotype, you can press that button there, clears that. If we've got two B alleles, it's B-type. Because this B gene says, or makes makes B-type sticks. And this gene makes B-type sticks. So you wind up with B-type sticks on the outside. So hence your B-type blood. Does that make sense? So the phenotype, the type of blood you are is what types of sticks do you have on the outside. In this case, because this one makes a B and this one makes a B, we have B-type sticks on the outside, so you're B. Good so far? It was the same story with the A. Now, what happens if you get an A allele and a B allele? Before I hit enter, let's think about this. The A allele is going to make an A-type stick and poke it on the outside. The B allele is going to make a B-type stick and poke it on the outside. So what types of sticks do I have poking on the outside? Oops, wrong button. I have both. I have A-type sticks and B-type sticking on the outside. One made A-type sticks. The B one made B-type sticks. So I've got both A and B-type sticks, hence I am AB-type blood. Does that make sense? Hence it's codominant. Because the A makes sticks, the B makes sticks, and they both show up. So it's codominant. Good so far? Okay. Now, let's ask about the O. If you are O genotype, think about what the cell was. So what was the O allele doing? Remember, the O allele was the broken version, so it doesn't make a stick. So if the O allele doesn't make any sticks, and this O allele doesn't make any sticks, what sticks do you have on the outside? None. You don't have any sticks. We call that O-type blood. So O-type blood is the lack of a stick. Does that make sense? Okay. So now, this is why there's a complete dominance thing between A and O and B and O. Because, watch this, if you're AO, the A allele makes an A-type stick. The O allele doesn't make a stick. So what sticks do you have on the outside? A-type sticks, hence your A-type blood. Does that make sense? Same thing if you have a BO. Not only do you smell bad, you have B-type sticks on the outside because the B-type allele contributes a B-type stick, and the O-type allele doesn't contribute a stick. So you wind up with B-type sticks on the outside, hence your B-type blood. So a BB and a BO both give you B-type blood. Same thing with an AA and an AO, both give you A-type blood. The only way to get O-type blood is to have two O's because that's the only way to not have any sticks on the outside of your cell. Does that make sense? Because either of the other two are going to make a stick. Now here's where the Punnett squares come in. Now that you understand the principle, you can do a Punnett square. So I've got a nice fancy dancy little Punnett square that you can use down here. So like if you've got a man that's AA and a woman that's OO, so she would be O-type blood. So he's A-type blood, she's O-type blood. Now there you go. Here's their 4 possible kids. They can only have A-type children. Cool, huh? Now here's the fun combo. if you've got, click Punnett Square, okay, here we go. If you've got an A, so they have A-type blood, and you have a B, they've got B-type blood, then you would have 25% chance of having AB-type blood, 25% chance of having B-type blood, 25% chance of having A-type blood, and 25% chance of having O-type blood. Fun, huh? And that's your phenotypic ratio in that case. So whereas in this case, let's try. Let's see here how I'm going to do this. Let's make this person, they're going to be an AB. So now look at this. In this case, I've got a person that's AA and a person that's AO. Both of those, I wouldn't report those differently because that's genotype. AA is an A-type phenotype and AO is also an A-type phenotype. So I would have 50% A. And then 25% AB and 25% B in this case. That's all it takes to do the blood typing questions. Now, the trick and the catch here, don't get deceived. There are two ways to be A-type blood, so check them both. They're not that tricky on the homework problem, but just for habits, I would recommend when you're doing that homework problem, I think it's problem number 4 on the homework. When it says they're an A-type parent, you don't know what their genotype is. You know it's either AA or AO, so test them both. Pretend they're both. So run a Punnett square with AA and figure out what the possibilities are there. Then run one with AO and figure out what the possibilities are there, and then sum all the possibilities together. So that problem is a process of elimination one, because you'll find that several of the parents can make more than one of the babies. So you're going to have to do a process of elimination. Is how you solve that problem. But anyway, you're welcome to play with this. Another fun feature here is if you've got the babies all figured out, then you can press, let's say I want to see what this baby's cell will look like. Then you can, sorry, my computer's being slow. Then you can press the display cell button and it'll put that baby up as opposed to over here. As opposed to over here, then you can put up that baby, etc. Okay, here we go. So that's how blood typing works. You're welcome to play with this spreadsheet all you want. Okay, the last couple of principles here. Multiple alleles. Actually, let me make a second video. This one's getting pretty long. I'll make a second video covering these last 3 principles.